Hi everyone, welcome to this video. This is the second part in our image processing and analysis in ImageJ Fiji course. I'm Stephen Cross, I'm from the Wolfson Bioimaging facility at the University of Bristol. In the first video, Don took you through the basics of image acquisition and processing with a focus on light microscopy. In this video, we're going to continue this and see how these principles apply to ImageJ and Fiji. We're going to do this using an example workflow. So what's going to happen is I'll talk for a few slides, going through some of the basics of ImageJ in general, the interface, and also how you load and save images and navigate around images once they're open in ImageJ. Then we're going to step by step go through core functionality within ImageJ, such as filtering and thresholding, and then see how this can be applied to this example workflow of segmenting cell nuclei and then measuring some properties and plotting that data. You'll find the worksheet and the example data at the GitHub link shown here. So it's github.com/wbif-bristol/imagej-fiji-level-1-course. First thing we want to look at is what exactly is ImageJ? Well, ImageJ is an open source Java-based image analysis platform. It was developed about 20 years ago by Wayne Rasband at the National Institute of Health in the USA. This is the ImageJ interface here. The one sort of major change that's happened recently, or the last few years, is the introduction of Fiji. So Fiji stands for Fiji is just ImageJ. It's core ImageJ, but it comes with a lot of additional pieces which are really useful, in particular for bioimage analysis. So plugins for example, you've got the bioformats importer, which will allow you to read in numerous proprietary image formats from companies like Leica and Zeiss and Nikon. You've got various segmentation and tracking functionality that isn't included in the base ImageJ package. There's also a really nice package manager, which we'll look at later on, which allows you to load in any of the sort of hundreds or potentially even thousands of ImageJ plugins which are available and have been previously published. If in doubt, just download Fiji. You can get it from fiji.sc. It's available on Windows, Linux and Mac operating systems. So just to go through some of the main menus here, obviously you've got the standard file open save menu common to a lot of programs, so I won't really dwell on that. You've also got the edit menu here, which again includes a lot of standard stuff, copy, paste. There are a couple of interesting features there, or functionality at this menu. One is the ability to invert the image intensity. Also dealing with regions of interest, which again is something we'll look at a little bit later on. In the image menu here, you've got the standard stack operations, so this is dealing with multi-dimensional images. You can also do basic image manipulations here, so scaling the image, cropping the image, zooming, you can rename it, and you can also play around with the way the image is displayed in terms of its colour. These are lookup tables. In the process menu, you're dealing with processing the image on a sort of pixel by pixel basis. So here you've got things like image filtering, ability to binarize the image and the binarization tools that go along with that, so filling in holes, generating outlines, skeletonization and so on. You can also do image calculations, so whether that's adding or subtracting or dividing fixed values, or adding images together, calculating the difference between images, and also the very handy background subtraction tool. Again, something we'll be working with during the workflow. In the Analyze menu, unsurprisingly, you've got things dealing with measurement. You've also got the very handy Analyze Particles function, which allows you to take a binarized image and identify individual regions from that and measure size, shape, also intensity of that object in other images. You can also do things like intensity profile measurements, and in Fiji there's co-localization plugins that you'll find in the Analyze menu. If you have extended ImageJ or Fiji in any way, you'll find a lot of that functionality in the plugins menu. If you've got ImageJ, that menu is going to look pretty sparse, but if you've gone with Fiji, you'll find many sort of subcategories there, such as tracking, segmentation. Down at the bottom here, you've got this context-sensitive bar. So as you're doing different stuff, this will just keep you up to date with what ImageJ is doing. So whether that's you're sort of moving your cursor around an image, it will tell you what XY coordinate you're at or what the intensity is. It'll also tell you the progress if you're running filters, things like that. If you want to select regions manually from your image, you can use these 6 or 7 buttons on the left here. So this is something we will be using later on, but you can draw like rectangles, ovals, or sort of more bespoke shapes, like freehand selection tool is there in the middle. You've got a magnifying glass and a grabby hand here for moving around the image, so zooming in and out and panning. And then just a little shout out to the lookup table button here. So we've already briefly mentioned lookup tables when on the image menu there, but there's a nice shortcut to lookup tables here as well. So first things first, we want to get some images into ImageJ Fiji that we can start doing some analysis on. The easiest way to do this is literally just to take an image file and drag it into the main ImageJ window here. So just pop it anywhere in here, and ImageJ is generally pretty good at determining from the file extension what sort of reading plugin it needs to use. So for example, we have a lot of like microscopes, and those generate these .lif files. If we drag a .lif in, it's going to recognize that it should be using the bioformats importer. Again, just to mention that that one's only available if you have downloaded Fiji. Otherwise, pretty much everything that we're going to cover in this session is compatible with both ImageJ Fiji, but here, yeah, file loading using bioformats is just a Fiji function. If you want to load in files a sort of more manual way, you can go to File and Open, and then select it from a browser there. You can also load in those image sequence files that you occasionally end up with where they've got a suffix, like a numerical suffix, so 0001, 0002, and so on. To do that, just go to File, Import Image Sequence, and it allows you to load in subsets of that or the entire set that you've got in your folder. And as I've already mentioned, if you've got proprietary formats, you can use bioformats. For this, you would go to File, Import, and then down here, Bioformats. ImageJ Fiji has often got a lot of sort of long menus, so you'll see these arrows top and bottom that allow you to scroll through. So when you go to this first time, you may not see bioformats, it's probably off the bottom, so you just go to the little arrow and scroll through. If you are using bioformats, you'll get presented with this window here. So this one here is just sort of control for loading. If in doubt, generally I'd leave most of these unticked. A few that are worth mentioning. So you can load in here some of the metadata. So this is just text data which is stored in the image file. So it can include just basic stuff, things like when was the image taken, potentially by what user, things like that. But with the sort of more advanced formats, such as like analyse or Zeiss CZIs, you'll get a lot more information such as laser powers and objective lenses and filters and so on. And if you want to look at that, maybe you've acquired your images and you can't remember how you, like what settings you used, or did you use the same settings across all images, you can load up these here. So just place ticks there, and it will open along with your image just a little window showing you what the metadata was. Now there's a couple of different metadata options here. The first one is this sort of standard metadata, and then the other one here is this OME XML metadata. This is sort of standardized data format, which makes it a little bit easier to navigate, although you will find that you won't get all of the metadata in the OME XML that is available just by looking at the raw data there. Something else that can be useful is this use virtual stack. So if you've got a computer that hasn't got a massive amount of memory, by that I mean the RAM, what you can do is use a virtual stack, and then ImageJ will only ever load in the actual frame that you're currently looking at. So maybe you've got a 50 gigabyte file and you obviously can't load that entire thing into memory. So what it does is it streams it dynamically from the hard drive as needed. Benefit of this is that it loads up really quick, but scrolling through that stack is going to be slower than if it was loaded into the memory because it's constantly searching for it on the hard drive. If you've got more than one image series within your proprietary format that you're loading with bioformats, you'll get presented with this window here where you can scroll through all the different series and then just put ticks against the ones that you want to load in. Previously mentioned the image metadata, so you've got the OME formatted metadata here, so it's all nicely sort of split down by categories, so images, pixels, channels, and so on, or you've just got the raw metadata here, so it's just literally just a text list. but it is complete. So it's all of the information that is stored within that image. Saving images, again, standard kind of stuff here. File, save as, and then you can select a format here. All typical stuff there. And then also you'll find some down here which come from whichever plugins you've got installed in your Fiji at the time. You can also save image sequences by going to File, Save As, Image Sequence, and then this allows you to export with a fixed number of like values at the end, and also what starting number you want to use. Now that we've got our image loaded into ImageJ Fiji, we're going to be presented with a window that looks like this on the right. So the image that I loaded as an example here is 4-dimensional. It's got Z slice, or multiple Z slices and 2 channels. You can have up to 5 dimensions in sort of standard ImageJ Fiji here. So if we had got a time element, along the bottom here you would have seen a little arrow, and that would have allowed us to progress through the different time series. If you want to just play that video, you can just click on the arrow and it would automatically go through. You can also press the backslash button and it would do the same thing. If you right click the arrow or go to Image, Stacks, Animation, Animation Options, you can change the rate at which the frames change over. You can also set whether or not the video goes on to a continuous loop. Okay, so now it's your turn to do something. What we're going to do is we're going to work through Worksheet Section 1. This one's just a nice easy intro. All we're doing is loading up Fiji, dragging in an image to have it import with bioformats, and then mucking around with the channels and slices sliders just so we can be confident that we can navigate around the image. If you haven't already done so, you want to download the example data.lif file from the GitHub and also the worksheet Word document. You can either do this by typing in the super catchy link here, github.com/wbif-bristol/imagej-fiji-level-1-course, or you could save yourself the effort and just click the link in the description below. The worksheet looks like this. So it's broken down into the different sections that we'll be working throughout this session. At the moment, we're just working on this one here, which is loading images. Common across all of these exercises is that in italics you've got a little description of what we're aiming to achieve, followed by the steps that you want to follow. The idea is not to have any hidden surprises in this, so it should be that everything you need to do has already been covered in the slides. One thing that you will find later on, especially when we get down to things like background subtraction, is that we're going to look at multiple different ways to achieve a goal. So the idea here is to sort of demonstrate how with image analysis and image processing, there's often no one correct way to achieve something. There might be lots of different ways which give similar results, but have, you know, advantages and disadvantages for that particular method. And it's up to you to decide which one works best for what you're trying to achieve. And that's really what we're sort of trying to highlight with this sort of branching pathways approach. Okay, so for worksheet section 1, it's just a case of loading in an image. So I've already got Fiji open here, and I've got my example data.lif file from the GitHub. I'm just going to drag that in, and as I saw on the slides, it automatically loads up the bioformats importer. One thing I didn't mention previously was that as you sort of scrolling through here or moving your mouse over, on the right hand side you're getting a nice text description of what each setting does. What I'm going to do is I'm just going to pop open the 2 metadata windows so we can have a look at those, otherwise leaving everything else unticked, clicking OK, and because there's only one series in this file, I didn't get that series selection. It's just straight away loaded up the image. So start with, we can see here some information about the acquisition, so different lasers that we had on the microscope. There'll be things like, say, lenses, physical distance per pixel, which we'll look at in more detail later on, but that's the sort of real world distance, in this case in microns for every pixel. And then also we've got the sort of raw metadata, and you can see here that there's a lot of information in here, most likely more than was in the OME XML formatted version. This is my window with the image, so I can scroll through in Z, so we can see some nuclei in blue in this channel, and some cells, again in this case being shown in blue. There are different lookup table options when you're loading in images with bioformats. The default here is just to colour everything blue. Okay, so now let's move on to some other basic functionality of navigating around our images. If we go to the Fiji window here, or the ImageJ window, we've got this magnifying glass and the grabby hand tool. So if you click the magnifying glass and then just left click and right click on your image, you'll see that you zoom in and out. You can also use the up and down keys on your keyboard, and that will do the same thing, so it's quite a nice shortcut. Similarly, just grabbing, select that left hand, and you can grab and drag around. One kind of handy tool is that as you're dragging around and you're zoomed in, you get this mini map. So that's the image, and then the little square inside just shows you whereabouts in that image you are. As I mentioned previously, ImageJ is capable of recognizing spatial calibration. As you move your mouse over the image, on the context-sensitive bit we saw earlier, you'll see the coordinates of where your mouse is, along with the pixel intensity at that location. If you haven't got a spatial calibration applied, maybe the image format that you loaded in just didn't support it, maybe you've loaded in a PNG, something like that, these will be in pixel units, whereas if you have loaded in an image from a microscope and it's been able to interpret the correct spatial calibration, or a spatial calibration in general, it will apply it here. And I say correct spatial calibration because it is important to make sure that what's being loaded is the true distance per pixel. Quite often you'll load something in from a microscope that maybe hasn't been set up right, or just hasn't been set up for spatial calibrations, yet is still outputting some information that Fiji interprets as being a spatial calibration. In that case, you might actually end up with incorrect measurements coming out. But if you know what the real distance per pixel is, you can set this by going to Analyse, Set Scale, and then you get this window here. So you just enter in a known distance in pixels, the known physical, like real world distance, and then you can also put in here units. You can also type in and ImageJ will recognize that you mean microns and change it to the correct Greek symbol. We can also use the brightness contrast tool to change how the intensity has been displayed on the image. So on this image on the right hand side, you can see that we've got some nuclei, but they're not particularly visible. This is because by default, ImageJ is trying to display the minimum intensity in that image at 0 as being black, and then the maximum possible intensity, so for an 8-bit image that would be 255, as being white. However, maybe the maximum brightness in that image is only 115. In that case, we're seeing pixels in the range sort of black through to kind of like mid-grey, but anything from mid-grey up to white we're not actually making use of. So what we can do is, if we go to the brightness contrast tool, so image adjust brightness / contrast, we can change what the minimum value, i.e. the black value, and what the maximum value, the white value, being displayed are. So if we stretch that, or sort of squash it down so that that full range of black through to white is being displayed just over the intensity range that we have in our image, you can see that we get a much better contrast. Now, one of the nice things about this tool is that just by changing this, it's only altering the way that ImageJ displays that information onto the screen. It's not having any fundamental effect on the pixel values as stored in the image. And that's very important, because as soon as you start mucking around with those, you open yourselves up to all manner of problems with being able to compare different images, maybe use different ranges, or maybe your measurements just stop making sense. So if you just simply want to change the brightness so that you can see it on the screen, you can change these sliders, close this window, and it's all good. If you should want to stretch those values so that the values stored within the image itself go to the full range of 0 to 255, you can do it by clicking apply. But as I say, you really want to treat that function with care, because then you're actually modifying your image, and it might make comparison with other images difficult or invalid. As Dom mentioned in the first video, images generally are just grayscale, so it's just a single channel with different intensity values between 0 and, say, 255. However, you might know that that particular channel was a DAPI stain, so that it should look blue, or it was a green stain, in which case you can change the lookup table. So this is simply the way that ImageJ is displaying that grayscale information to the screen. So you can do this by going to the LUT button on the main panel. and selecting just standard colours. So in this case blue, we can see our nuclei in blue, or we can just change it so the nuclei appear in green. You can also change it to have additional benefits. So for example, this one here is called the high-low lookup table, HILO. And what that does is it displays any pixel with the minimum value as blue and any pixel at the maximum value as being red. So this is a very quick way to see whether any pixels in your image have been saturated. Alternatively, what you can use is something like the fire lookup table, which rather than just going from black through to, say, yellow, goes to a few different colours. So in this case, it goes from black to pink, red, orange, yellow, and then through to white. And with that additional variation, you can start to pick up some features in the background that you wouldn't be able to see otherwise. So for example, here we're able to pick up these tiny little flecks in the background that are maybe visible in the green, and certainly to me, I can't see those at all. Even this feature here is starting to disappear in the blue, whereas in the fire lookup table, it's very easy to see. Also worth noting that, again, image data isn't being adjusted by anything when you set the lookup table. This is literally just ImageJ's way of displaying it onto the screen. So now we're going to go on to Worksheet section 2. This one is simply about taking that image that we've already loaded into Fiji, changing the brightness contrast, zooming and panning around the image using the tools we saw a few slides ago, and having a look at the effect of a few different lookup tables, and maybe even just leaving it on one that you think is useful. For Worksheet section 2, we're just taking the image that we loaded in the first section and starting to change the brightness and contrast, play around with different lookup tables to get a feel for what we like, and also just to use the pan and zoom tools to have a closer look at the image. So first things first, we want to find a slice which has got relatively decent focus. So if we look here, even though the image itself is not particularly clear to see at this point, we can see that we're able to get most of the nuclei in focus. So what we want to do is go image adjust, brightness and contrast, and we can just hit auto here, and it scales itself to not quite the maximum of the displayed range. I think it's like 99% of all pixels are within this range or something similar. We can change this manually using the minimum and maximum sliders. So the minimum here, as I mentioned previously, just changing at what value black starts. So here, anything below now 34 is being displayed as black, irrespective of its value. I'm actually just going to set that down at 0, but then changing this maximum value. So as you see, we're pulling this down more and more, just being displayed as bright blue. So if we leave that there, that's good. And I'm going to close this because I don't want to fundamentally change the pixel values in my image. We can change lookup tables a couple of ways. We can either do this the sort of long route, which is to go to Image, Lookup Tables, and go here, or we can just simply use the Fiji shortcut LUT here. And just by switching to grey, you already get a better view of the image. So I find blue's not particularly easy to see, especially for those dark shades. I wasn't able to make out this fine detail here, whereas by switching to grey, it kind of pops out much better. We can also change to some of the others that we were talking about. So high-low, this one here, is displaying anything that is saturated on the current display range as red. So the key thing to note here is that this is just based on the display range. So if you want to know if any pixels are actually saturated in your image, you set that to the maximum value. And as we go through, we can see that a couple of these cells, which are presumably dying, the nuclei there are saturated. But fortunately for the analysis, we're going to be removing those anyway because we don't want to analyze them. But I'm going to set this back down here and then actually leave it on the fire lookup table because I quite like this. I think that by going through this different intensity or the different hues, it does make all the background come out that much easier. The final thing we want to look at is panning and zooming. So there are tools. So this is it, left click to zoom in, right click to zoom out. And you can see we're getting this little map as to where we are in the image as we're doing that. And the pan tool there. And we've got shortcuts, so keyboard up and down to zoom in and out. And also we can hold down, even if we're not in either of these, we can hold down on the spacebar on the keyboard and that automatically puts us into pan mode so we can move around. The next thing we're going to look at is changing the stack itself. So right now all we've done is loaded in that image and started moving around in it. But what if we want to pull out different elements or indeed split down the different channels of that hyperstack? So in order to pull out the different channels, we can use the split channels tool at image, color, and split channels. If we decided that we wanted to merge those back together, or if we'd got different sources that we'd loaded in as separate images and wanted to merge together, we can use the image, color, and merge channels tool. Now, when you merge the channels, you'll see that they'll end up in a different or slightly different format to when you split them down. So originally, when we loaded in the images, they're in what ImageJ considers to be color mode. And in this, the different channels are displayed one at a time. Whereas when you've done the merge channels, we've got create composite enabled. So what it will do is it will display all of those channels simultaneously. You can go to image, color, and then channels tool to change whether or not it's being displayed as grayscale, color, or the composite. Another handy feature of ImageJ is being able to duplicate stacks. You could do this with image and duplicate. This is going to be useful for the worksheet because as we're going through, it's useful to either make physical sort of file saves or just duplicate the stack before you apply filters and thresholds in case you decide that what you've done you don't like and you want to go back. Other bits that are useful are the ability to just rename the images, so that's an image and rename, and also to be able to extract subsets of your hyperstacks or your 3D stacks. You can do this with image stacks tools make substack, and you'll end up with a dialog which looks a bit like this. The number of input fields there will vary depending on the number of dimensions of your image. For us, we've got 4-dimensional image, we've got 2 channels and 25 slices, so it looks like this. And you could pull out, say, the first channel and only slices between 10 and 20. Something else we might want to do is to take a 3D or multi-dimensional stack and project it down by removing one dimension, typically the Z dimension. So what this would do is this takes, if we consider a 3D stack of nuclei that we're using for the worksheet, it will take a statistical value at each XY location on that image based off the entire stack. So if we look at the max intensity projection here, at the top left location here, the pixel is the maximum value that was present at that location in any of the slices. So what we end up with is a nice sort of projection of all those nuclei reduced down into a single 2D image. Now, as you'll see on the worksheet example data stack, those nuclei do appear over sort of 3 or 4 different slices. So by doing this max projection, we're able to effectively compress them all down into a single plane that will make subsequent analysis easier. If you have objects which appear above and below each other in the Z plane, all you're going to do is just sort of squash them down onto each other, and you can actually make things difficult or more difficult for yourself, which is maybe where taking the substack like we saw in the previous slide beforehand would be useful. Similarly, we're able to extract out the background signal profile by doing a minimum intensity projection. So at each location, it's not considering now these bright nuclei here because there's always a darker pixel behind them. These tend to come above and below in the stack where the nuclei aren't present. So this is useful for pulling out things like background, but it doesn't always work. Again, if the nuclei were present in every single slice and we didn't have the sort of really out of focus regions above and below, you would still start to see the nuclei, which you can, if you look closer, you can see the outlines of nuclei here, but they're a lot lower than they are in the max intensity projection. And then just giving a couple of other examples here, like taking the mean of all pixels at each XY location or taking the standard deviation. So now we're going to look at worksheet section 3. The aim here is to take that 4-dimensional image that we've got, split out the 2 channels, and then just dump the second channel that contain the cells. For the worksheet we're doing here, for the entire sort of workflow, we're not going to consider anything in channel 2. So then taking that now 3D stack of the nuclei, we want to generate a couple of images. One of them is just going to be a Z projection, and another one's going to be a single Z slice, so a single slice through the image. And this is the first of these sort of like branching pathways for this worksheet that I mentioned earlier on. So it's up to you to kind of decide at the end of this which one you think looks better. Okay, so now what we want to do is to extract out a couple of example images showing all the nuclei. And the key thing here is that when we say sort of a best image of the nuclei, for the subsequent segmentation, what we want is bright nuclei which all appear in focus at the same plane and also have nice sharp edges, because it's those sharp edges that are going to make things like thresholding a lot easier later on. So the first thing to do is to split this down into its two channels. So color, split channels. So now what you'll see is we've got those two channels, and ImageJ has automatically renamed them, so it's C1 and C2. So this is the one that we're not interested in. This is where the cells are. So I'm just going to remove that, and this leaves us with our full 3D stack showing the nuclei. And you can see how they appear in focus over a range of Z slices. So the first thing I'm going to do is to generate a maximum intensity projection. You can also specify a range over which you want that max projection to be calculated. I'm just going to do it over the full range. So click OK there, and you can see this has done pretty much what we're after. So we've got nice sharp outlines. So I go to Image, Rename, and I'm going to call that Max IP, Max Intensity Projection. I then go back to this image, and the next thing I want to do is to decide which slice of this has the best focus. I would say here, and we can see as we're moving around that it's updating this information here, so it's telling me that this is currently slice 9 out of 25. So if I go to Image, Stacks, Tools, Make Substack, and if I type in 9, it'll give me that substack. And again, I'm going to rename this so that my workspace is tidy, Substack. There are a few different ways you could have achieved this exactly the same thing. For example, we could find that, and then we could go to Image and Duplicate. Also worth noting in Fiji and ImageJ is that there's a bunch of keyboard shortcuts that get listed in the menus. So I could just use Ctrl-Shift-D here to access Duplicate. And if I don't have Duplicate stack enabled, it would just give me the current image that I'm looking at as a new window. So that's exactly the same as we've achieved there. So two ways to achieve the same thing. I've now got three windows. So I've got the max IP one, I've got the substack, and I've got the full 3D stack that we're still going to want. I now want to choose which of these two I think looks best. So for me, I would say it's the max intensity projection. The reason for that is that every nucleus here is in focus, whereas if you look at that substack, unsurprisingly, there are a few cases, especially around here and around here, that you can see you're losing that edge sort of contrast, whereas here they're nice and sharp. So I remove that, and now we can go on to the next section. Now that we've got our picture of the nuclei, what we want to do is to optimize it by removing some of that background. The background we're talking about here is that long length scale variation in intensity that we're seeing. It could come from out of focus objects. Actually, in this case, it's all fake. I put it on there just as a demonstration because actually the image looked quite clean to start with. But for the purpose of removing that fake background, what we're going to do is we're going to look at a couple of different approaches. The first of these is ImageJ's rolling ball background subtraction. So you get to this by going to Process and then Subtract Background. And effectively what it does is it rolls an imaginary ball along the underside of your intensity surface. So if you imagine your surface in 3D, so obviously you've got X and Y, and then the Z dimension being the brightness of each pixel. So getting higher, the brighter it is. It's easier to visualize this in one dimension. So imagine here that we've taken a cross section through the image, and what you can see is you've got this long length scale variation here. So this is the background that we want to remove. And then you've got the nuclei and spots and things like that superimposed on top of it. So that's what these sharp spikes are here. And what the rolling ball does is, as long as the rolling ball is larger than the features you're interested in, it's able to trace out that long length scale background, but it can't trace out these sharp spikes. So in this case, we would choose a rolling ball maybe of radius 10 or 20 pixels. And you can see this here just rolling underneath the image. And see how that traces out that background profile here, but doesn't include these spikes. And then the one problem we've got at the moment is that this is offset by a certain amount. So if we roll that ball back over the other side, over the top of that background surface, you can see now it superimposes nicely upon the background. And then ImageJ can just subtract this red line from the initial line, and it would just give us our spikes, or in 2D, the nuclei that we're interested in. This works for when you've got bright objects on a dark background. The rolling ball option has this light background option. So if we had dark features on a light background, like you might in, say, a bright field image, you would enable that, and it effectively just rolls the ball from the other side. The subtract background feature also allows you to select whether or not you just want to create the background. So in that case, rather than automatically subtracting off the calculated background, it just gives you this background image. and the image would look something like this for our images. So this is the rolling ball here with a radius of 50, so you can see how it's traced out that long length scale features. There's another option here which is sliding paraboloid. This is just a different sort of physical model, so rather than being a ball it's a paraboloid, and you can see most noticeably at the edge often you get different sort of fade outs. And you can also enable previewing here, as is common with a lot of ImageJ features, so as you're changing these settings you can see in real time what the effect's going to be. The other way you might want to subtract background is if you've already got a background image. So this could either be that you've run the rolling ball and you've generated that background image, or as you'll see in the worksheet section 4, we might generate a minimum intensity projection and subtract that off our maximum intensity projection to achieve the background subtraction result. To do image calculation, you go to Process and Image Calculator, you specify the first image, in this case this would be our single image, and then we can set the operation here to subtract and then select off our minimum intensity projection. There are a whole load of other operations here, so obviously like adding, multiplying, dividing, but also calculating differences, calculating the maximum value between two images, and so on. It's also worth noting the 32-bit float result. So far we've just been working with integer intensities, so values that go, in this case, on an 8-bit scale between 0 and 255. However, certain calculations like divisions and subtractions can result in fractional pixel intensities or negative pixel intensities, which would be rounded or set to 0 respectively with the 8-bit integer scale. So by enabling 32-bit float result, it actually captures each pixel as a floating point decimal. Now for worksheet section 4, what we want to do is again take two different approaches to subtract off that background. One will be taking our previous max intensity projection image and running the rolling ball subtraction. The other will be going to the stack, generating a minimum intensity projection, and then using the image calculator to subtract that minimum intensity projection from the maximum intensity projection. As before, we're then going to look at the results from those two operations and decide which one we think has given us the best image. And in that case, it's just bright nuclei, sharp edges, and complete background subtraction as possible. Right, so exercise section 4. What we've got so far is we've got our original stack, just showing the nuclei, and we've got our maximum intensity projection image. First thing we had to do in this exercise was to generate a minimum intensity projection. So we go to Image, Stacks, Z-Project, and then now change this to min intensity. So you can see here that this really shows up that background intensity profile. And don't worry by the fact that it looks different to here. This again is just the display range. So if we go to Image, Adjust Brightness Contrast, we can set it to anything, and there you go, see that. It looks about the same. Fact is the pixel intensities aren't any different between the two because we're not hitting apply. So it's just about how it's being portrayed on the screen. I want to just rename this for the sake of organization, so I'm going to call that min IP. And now we're going to run the background subtraction using those two methods we saw in the slides. So in order to do that, I'm going to duplicate this image. So using the keyboard shortcut Control-Shift-D, I'm just going to call this rolling ball. What I'm going to do here is select that image and then go to Process, Subtract Background, and now enable preview so I can see in real time what's happening. So with a rolling ball radius of 50, I can still see there's a lot of this background that hasn't been removed. So maybe I'll remove that down to 40. It's still there, so I'll just keep going. Still slight bit there. 20. So that looks good. We're starting to lose a little bit of intensity on the nuclei at the edge here. You can see that as I turn it on and off. And if I go much below that, then I'm going to start losing these nuclei entirely. So if we go down to say 5, turn the preview on, so you can see how the nuclei now are looking messy, even at 10. So I think 20 was a good compromise between removing the background but not affecting the nuclei too much. And we can also test the effect of sliding paraboloid. So as I say, this often has an effect right at the edges of the image. So I'm going to use a sliding paraboloid here because it does a better job at subtracting off the background from this nucleus here. So that's good. So that's one of my background subtractions. The other one's going to be using the image calculator to subtract off the minip from the maxip image. So I select maxip, go to Process, Image Calculator, and then select Maximum IP, subtract, minip. Because of the way this works with that being a minimum and maximum intensity projection from the same image, I know that I'm not going to be able to get negative values because there's no value in this that can be smaller than the values in this. So I don't need to enable the 32-bit float result. So I can just hit create, and then that's generated an image like this. Because of the way they work, the offsets are now slightly different. I think the maximum intensity projection subtracted the minimum intensity projection possibly looks a bit better. We don't have so much background, although the thresholds that we'll be applying later on don't really mind about having a little bit of offset. However, just for the sake of having to choose one over the other, I'm going to retain this one. So now I can close the minip, maxip, and rolling ball, and then I'm just going to use this one going forward. So far we've looked at selecting subsets of the image in terms of like substacks, but not of a single plane. So now that we've got that single plane showing the nuclei against a flat background, maybe we want to identify manually some of those nuclei. One reason for doing this is to get a rough estimate of how large the nuclei are, so that subsequently we can remove anything which is massively outside this range. So really small things that might correspond to noise, or really large things that are maybe clusters of nuclei that we can't accurately measure. In order to do this, we can use the region selection tools which you can find on the main ImageJ panel. So I've shown 6 on the right hand side here. You've got the rectangle tool, you've got this oval tool, then you've got this segmented line tool. So here you just click on every node that you want, and ImageJ will draw a straight line between, and then when you right click it will finish off drawing, and then it will link up the final point to the first point. Or you have this freehand selection tool where you just literally hold down the left button and draw what you want. Similarly, while these two are enclosed regions, i.e. the last point always joins up to the first point, you have just section line tools. So in this case you've got the segmented line, so when this ends it doesn't join them up, and so whereas here we're defining an area, with this one we're effectively defining an edge. Similarly, we've just got the freehand line tool. So for what we're doing here of measuring nuclei, you wouldn't use these two. You can also do the Magic Wand tool. So what this does is it identifies contiguous regions of pixels with similar pixel intensities. So the way we use this is we select this Magic Wand button here, and then click somewhere within our object that we want to identify. So here you can see I've selected this bit, and what it's doing is it's all pixels within tolerance of the same intensity value. So as this increases, we're effectively saying all values that are within currently 65 intensity values of the value of this pixel are incorporated into this region. So you can see how this grows out. By the time we get to around a tolerance of, for this one, about 70, we're nicely, or even 50 here, we're identifying nicely the boundary of that nucleus, but then as we allow an even greater tolerance, that value actually incorporates the background, and so we just end up selecting the entire image. The way it works from a sort of technical point of view is that it draws a straight line from going right from the point you've selected until it finds an edge, and then it just follows that edge round clockwise. As such, if you're selecting an object, it's best to select either just inside the object on the right hand side here, or just outside the object on the left hand side there. Once we've got a region, we can start making measurements. So in order to tell ImageJ what measurements we want to make, we can go to Analyze and Set Measurements, and you'll get this dialog up here. So by default it has things like the area and I think the min-max grey value, and that's about it. However, you can enable things like centroids and medians. So a lot of these are just intensity values, so you don't actually have to have a region selected. If you don't have a region selected, it will just measure the entire image. However, there's also a lot of shape metrics here, so things like the perimeter, the centroid, center of mass, things like ferrite diameter here, which only work if you've actually got a region selected. Another useful tool that we'll be using later on is the ability to redirect to. And the reason for this is that if you are using the analyze particles function, for example, it will identify a single particle, a single object, as a continuous region of black pixels or zero intensity pixels, depending on how ImageJ is set up. Now, if you were to make intensity measurements of that, it would always tell you that your mean and min and max intensity values are just 0, and that's not very useful. So what you can say is, I'm going to detect an object in this image, but then I want ImageJ to take the intensity measurements from another image. And that is where you would use redirect to. You would select the image you want to take intensity measurements of those particular regions from. To take basic measurements, you can then just go to Analyze and Measure, and doing so you will end up with a results table. So in the results table, each row here corresponds to one sort of measurement set. So if you had an object selected, one row there would be the measurements corresponding to that particular object. In here you can see that I've got measurements for area, mean, minimum and maximum intensities. If you've got the results table, you should also have an option here called results, and that will give you some additional settings, so you can calculate the summary. So that's just the minimum, standard deviation, maximum and mean of all columns, and those will appear as four rows at the bottom. You can also plot a histogram using the distribution function. You can sort based on one column, or you can plot two columns against each other, either with connected lines or isolated points, and include things like error bars with that. But we'll be looking at the distribution and the plotting functions a bit later on, once we've actually started segmenting our nuclei out. One thing to note is that if you have a kind of results table, so something in this format, but it's called anything other than results, you won't have this option here. So if you end up with a different window, you can go to File and Rename in this window, rename it to simply Results, and then you'll get these additional options. One other thing that's worth a shout out at this point is the Linear Intensity Profile tool. So if you draw a straight line or one of those segmented lines, you can then extract the intensity profile along that line. So it was with this tool that I generated the profiles we were viewing the background subtraction using rolling ball with. So when you go to Analyze and Plot Profile, it will give you the plot window that looks like this. So you can see here that the intensities at either edge are low, so that's corresponding to these regions here, and then they peak up in the center around these bright regions there. ImageJ's got a fine plotting tool, but if you wanted to extract out those coordinates to use in another piece of software that maybe you're more familiar with, you can hit List and you'll get a results table here showing XY values. And because this window, even though it was results, wasn't called results, as I mentioned on the previous slide, you don't have that results option. Now that we've got our regions, we maybe want to store them for use later on, or possibly even just transfer them over to a different image. So for that, we can use the Region of Interest Manager. Once we've got a region selected on our image, we can add a region of interest by going to Edit, Selection, Add to Manager, or hitting Control-T on the keyboard. What this will do is it'll add it to the manager, so you'll get a new entry here. You can click these to re-enable them on the image, or you can transfer the regions to another image by opening up or selecting that other image and then hitting the relevant region there, and you'll see that it appears in that image so that you could move it around or take measurements or crop it out, anything like that. You can also use the Show All button here to display all regions that have been selected on the image simultaneously. Other features are once you've got your measurements there, once you've got your regions here, you can make a measurement of one or all of the regions simultaneously. So this is similar to going to Analyze and Measure, but it just does it for all of those regions, so that makes that kind of analysis a bit quicker. And also, if you've got these regions and you want to store them for later, you can go to More and then use the Save or Load options to store them or load them from File. Right, now we can go through to Worksheet section 5. So what we're going to do for this is just try and make a few characteristic measurements of nuclear areas so that we know roughly how big those nuclei should be in terms of the calibrated units of the image, so microns squared. I think we'll make 5, 6, 7 measurements maybe, add those to the Regions of Interest Manager, then use the measure function to measure them all simultaneously, and use the results table summarize function to get the mean nucleus area. Okay, so what we want to do is to select out maybe 5 or so nuclei, sort of characteristic size nuclei, and see what their areas are. So I'm going to do this using a few of the different tools, but first off, I just want to zoom in ever so slightly, make my job a bit easier. So for one, maybe we'll use the oval tool. So do this here, and it kind of fits, but it's not perfect, but it gives us a rough approximation. So if I go to Edit, Selection, and Add to Manager, you see the Region of Interest Manager here opens up, and then we've got that. So if I were to select somewhere else in the image, so to just remove that selection, I can now re-enable it here. I might also use the segmented line tool, so let's do that one here. And then just right click to finish. It's a bit wonky, but here we can drag these nodes to where we want them. And again, I can press Control T now, and I've added it to the Region of Interest Manager. So you can see how I can switch between the two. Let's try a freehand. A bit wonky, but it does the trick. Add. Also, a lot of these you'll see I've got little red arrows here. So if I right click, it lets me select a slightly different one. So whereas with the oval selection, it was constrained along X and Y. If I right click here and go elliptical, it does it like this, so I can get these that are on a bit of an angle a bit better. So let's go for this one here. Allows me to just fit that one a bit better. T, and then I'll make one more, again using the freehand. Oh, in fact, no, let's use the wand. So I'll select that there, and then if I double click on the wand tool and then increase that tolerance, see how it's slowly filling out until around there. I'm pretty happy with that. Let's go to 44. Okay, so if I hit show all, it shows all of those, and I've got labels enabled, so you can see that they're numbered. So 1, 2, where is it? 1, 2, 3, 4, 5. Or I can knock the labels off and I just see the outlines. I can also Control A on my keyboard here to select all of these, and then just hit measure, and now it opens up a results table with the measurements that I'd previously selected. So I'd done that by going to Analyze and Set Measurements. In mine it was already enabled as area, should be enabled by default within ImageJ. And then I just want to calculate the average area. So if I go to Results and then Summarize, you'll see we've got an additional 4 columns here, or sorry, 4 rows, showing the mean, standard deviation, minimum and maximum. So I can see that my mean area is around 175 microns squared. We'll just sort of make a mental note of that, because in a couple of exercises time we're going to want to use that. The next thing we're going to do is to apply some spatial filters to the image to remove some of the noise that comes from the detector, and also just uneven distribution of fluorophore within the sample. We can also get rid of little background artifacts, just little speckles, because otherwise when we do the automatic detection, these will be picked up as small objects. And while we can filter these out using size filters, and that was why previously we were estimating the size of the nucleus, we want to minimize the amount of errors and artifacts in the image before we do that automatic detection. So you can get through to a lot of standard filters by just going to the Process Filters menu, and then you'll see them all listed out. So on the right hand side I've summarized a few of these. You can see in top left this is just an unfiltered image of a nucleus, and then below that I've applied a median filter with a range of 2 pixels. If you're unsure what all these different filters mean, probably worth going back to the first video where Dom talks in detail about spatial filtering. So the nice thing about the median filter here is that it's edge preserving, so that it, even though it's smoothed out the texture within, it's retained this nice sharp edge, and it's that sharp edge that we're really interested in preserving for the thresholding. Alternatively, you can apply Gaussian filters, which do a good job at removing noise, but as you increase the range on this, you can see the edge of that nucleus blurring out. So it is going to make segmentation of the nuclei, especially if you've got 2 close together, increasingly difficult. The Process Filters menu also includes a range of other filters, such as Sobel filter, which enhances edge objects or edge features. So you can see here this bright ring with some smaller ridges here where the texture changes within the image. You can also specify your own filter if you want, if you're referring to a paper maybe and they've specified a particular kernel, although typically everything you'd want would be within the standard filters that have been applied. You can also find another few. So process and smooth and process noise are actually just shortcuts to the mean and median filters that you would find in the filters menu. both with range or sigma of 1 pixel. One filter that is worth mentioning that I haven't looked at so far is the remove outliers. So this is a process, noise, remove outliers. And this is effectively the same as a median filter, but rather than applying the median filter to every single pixel, it only replaces those pixels which are above a particular intensity threshold with the median value within the local range specified by radius. So if we have this raw image here, I put raw in inverted commas because I manually put in a bit of sort of salt and pepper noise here. So if we apply this with a radius of 1, you can see that it's removed these tiny outliers here, but this larger object has only been reduced slightly. But if I increase that to 2, you see that all of those large features have gone, whilst the fundamental intensity distribution within that nucleus remains largely unchanged. So now we can move on to section 6 of the worksheet. All we're doing here is we're taking that optimized image that we got to in section 4, where we've got the nuclei in 2D against a sort of relatively low intensity background. We're just going to duplicate that image and apply a few different filters to see which of these gives us a nice sharp nuclear edge with a reduction in noise. Okay, so what we're going to do now is just test out a few different filters on this image. What we want to do is to remove this kind of speckly noise that we're seeing here, maybe some of this speckling in the background. Also, if we really enhance the contrast here, you'll see the detector noise. We want to reduce that because if we were to apply a threshold, you'd see that you get quite sharp, jaggedy edges, whereas we would like a nice smooth edge to our segmented nuclei. So first thing we're going to look at is I'm going to try a Gaussian. So I could duplicate the image at this point, or I can just go and use the preview tool here. So within each of these filters, you're able to see in real time how the changes are being applied. So with no filter, with a radius of 1 pixel, with a radius of 2 pixels. So you can see very quickly the edges of these nuclei are blurring, and that's going to make segmentation difficult, especially for these which are close or already in contact, because those are going to get identified once we thresholded, probably as a single contiguous object. Other options, median is generally very good. So this is with a radius of 1. You can see here that we've just got rid of a lot of that speckle, but are retaining these sharp edges. So we can put that at 1 or even 2. We'll leave that for the moment, may well come back to that. Another filter which we haven't looked at previously is the variance. And although it's not necessarily that useful for what we're doing here, so if we look at this, just calculating the local variance, it is useful if you're doing processing of like bright field images where you've got different intensity structures. So sometimes they'll be darker or sometimes lighter than the background. Just pulling out their kind of texture through a variance filter will allow you to highlight the structures of interest and then apply thresholding. But for now, I'm going to go with a median filter, and I'm going to put that at a radius of 2 and get something like this. Now, actually, before that, what I want to do, so I've just undone there using Control-Z, is I want to make a duplicate because I want an original copy of that image now, that when we do our automated analysis of the nuclei, I want to be able to take intensity measurements from that one using the redirect to function. So we've got this, and I'll just call this as edit and rename, or image and rename. I'll just call that intensity image. And then this one here, I will rename as binary, because ultimately that's going to be what happens to it. So the next thing we're going to look at is intensity thresholding. And this allows us to classify our image into pixels which have intensities greater than that threshold, so are considered to be foreground pixels, and those below that threshold which are considered to be background. Now, one way to do this is to set the threshold manually using sliders. So this is image, adjust, and threshold. And you move that slider, and what you see in real time is that you get red labeled pixels, or pixels identified as red. These are the ones above that threshold, which would be considered foreground. So when we've got only 5%, so it shows the percentage here, so when we're 5% over the threshold, you can see that we're actually missing a lot of the pixels corresponding to that nucleus. 15% over, in this case, looks about right, and 25 is too high. We're getting a lot of the background haze around the nuclei being detected, as well as a lot of this background noise. So we would go here for something around 15% over. However, setting something manually like this is time consuming. It's not particularly good for our automated workflow. If we've got 10,000 images from some high content system, you don't want to be setting you don't want to be setting manual thresholds, and also just reproducibility. It's very prone to user bias. So actually what you would typically use would be an automated threshold. You just go to Image, Adjust and Auto Threshold, and you'll see this window here. You can set it to either be white objects on a black background or vice versa, and you can select a different automatic thresholding algorithm that it's going to apply. So there's about 16 different automatic thresholds in this plugin. The best thing to do is to take your image, try all, for which is the first option, and then you will get an example image that looks something like this. So with an input image like that, it gives you the result of all of those different thresholding algorithms, so you can see which one has worked best for you. And if you were to zoom in, you can just about make out that there's names below each one. So then when you go back to the plugin, you just select which threshold you wanted to use, click OK, and then it applies it. So the benefit of this is that it's automatically selecting where that threshold should go, so it's removing user bias, and it's also just making the whole process a lot easier because you don't have to manually set it on an image by image basis. Now this threshold is a global threshold, so it's the same intensity value being applied for every single pixel within the image. However, you might have some images where you've got an intensity gradient. Maybe there's some background which you can't remove, so one edge of the image, all the features are brighter than at the other edge. If you applied a global threshold to that, you would probably miss off a lot on the darker region, and you might start segmenting the background in the brighter region. So what you can do is you can use the local threshold function, so image, adjust and auto local threshold, which works in basically the same way, but it applies different intensity thresholds at every pixel of the image, and those thresholds are calculated locally up to the radius specified here. Nice thing about the auto local threshold is that it has the same feature of being able to try all. It does take a little bit longer because rather than just batching all the pixels in one go, it has to locally calculate it on a pixel by pixel basis. But yeah, if you've got a range of intensities and the global thresholds just aren't working for you, it's worth checking out auto local threshold. So now we're on to worksheet section 7. The idea now is to take the filtered image that we got in section 6 and test out a few different thresholding methods. Use the auto thresholding tool, and then once you've decided which one you want, generate a binarized image of that. Now don't worry at this point if you end up with a few holes or speckles. The next section we're going to look at is optimizing these binary images. To binarize our image, we're going to use the auto threshold tool. So first off, we select the image that we've previously filtered and already renamed binary, and then go to Image, Adjust, Auto Threshold. By default, ImageJ assumes that objects are black on a white background. So we want to disable this, so we've actually got black objects, white background, and then we're just going to leave it at try all. So we can see the different results here, and we can use the up and down on the keyboard to zoom in on different ones. And you can see the names below here for each of the different threshold algorithms. Looks like Huang's done a pretty decent job here. There are a few cases where adjacent nuclei have been merged together, but as I said in the brief for this particular exercise, that doesn't matter so much because the next step is going to be optimizing these images, like splitting these down. Others have done a rubbish job, so intermodes in this case doesn't work. There will be cases where intermodes is really good. It's one of the cases where you really just have to run try all and see for these particular images which one works best. Otsu is often a good one, although here you can see that it's not doing a great job with these lower intensity nuclei at the bottom. So I think we're going to go with Huang. So we can just close this and then go back to our image, image, adjust, auto threshold. Now we select Huang, again disable that, and now we've got a binarized image, black nuclei on a white background. Now that we've binarized our image, we just want to tidy it up a little bit. This is mainly things like filling in holes and also splitting adjacent nuclei which have become merged together into a single contiguous unit. In the process binary menu of ImageJ, you'll find a whole load of different binary operations, 6 of which I've listed out on the right hand side. So if we take this unaltered image here, so this is a sort of nucleus with a few holes in it, What dilate will do is it goes around and finds any pixel which has been identified as being background, and then if it's in contact with at least one foreground pixel, so a black pixel, it will itself be converted into a black pixel. So you can see here that a lot of these, which are just single pixel wide, all get converted into black. However, this bit down the center here, which is slightly wider, you do still get a few pixels which are identified as being background. We have a road which does just the opposite operation. So here, any foreground objects, any foreground or black pixels that are in contact with at least one white pixel will be converted into a background white pixel. There are open and closing operations which aren't listed in the on the right hand side here, but are available in that menu. And all this does is it does a sequential dilate a road or a road dilate step, which has the result of closing in or opening up the holes. There's also a fill hole step here, which will identify any completely enclosed background region and set it to foreground. Outline does pretty much what you would expect. And there's also this skeletonized tool here, which reduces this binarized object down to a single line. And that's useful if you're looking at things like DNA or microtubules, anything that's sort of extended and maybe you want to measure lengths of. We've also got a distance map. So this is slightly different in that you don't get a binary image out of it. You actually get an integer image with multiple grayscale levels. So here, every pixel is equal to the distance of that pixel from the edge. And actually the lookup table here has been inverted, so it looks brighter at the edge than it is in the center. But what you would actually find if you looked at the pixel values here are that these are large and they correspond to how far this pixel was from the background, and they decrease in value as you go outwards. Another important thing to note is that there are options for binarization processes. So if you go to process binary and operations, you can do things like setting the logic for whether black objects or black pixels are identified as being object or foreground. And you can also set the number of iterations that this uses. So for example, if you wanted to do sequential dilate steps, so not just taking into consideration one pixel, but then have it run through multiple times to dilate even further, you can set that there. One extra binary process that ImageJ supports is watershed segmentation. With watershed segmentation, we're taking objects that look like this, where you've got multiple interconnected lobes with narrow necks between them, and it's going to split them down, because we're assuming here that these correspond to three objects which have become merged together in the pre-processing steps. So by drawing dividing lines, when we subsequently run analyze particles, they'll be identified as three separate objects. The way the watershed segmentation works in ImageJ is to use a distance-based watershed. So it calculates the distance transform that we saw in the previous slide. So this is its non-inverted form. So brighter pixels at the center corresponding to larger distances to the background, and you'll get three local maxima here. So for the logic of how people think of watershed, we invert this, so effectively these become three basins. And if we were to take a line section through this, you would end up with a profile that looks something like this in blue. So we identify the local minima, and we assume that these correspond to basins, and that we fill these basins up with water, such as they're filling like this. So at this point here, where in this schematic we're at a level of 40, you can see two adjacent basins are being filled up individually or independently. And then as the water level rises, we reach a point that two adjacent basins, which have been labelled differently, come into contact. At that point of contact, ImageJ would draw a dividing line between them so that they remain separate. You can also see at this point that we've now started to fill up a third basin, so this would correspond to this cell here, and as the water level rises, so now by H160, we again have to draw a dividing line because the yellow and pink basins have come into contact. What this looks like in reality is that before watershed, you've got a few cases where you've got nuclei which have come into contact, such as here, here, and here, and then following watershed, it's just drawn a background line between them, so then they're now going to be identified as separate objects. Now we're going to go on to Worksheet section 8. All we're doing here is we're taking the binarized image from section 7 and testing out a few of the different binarization processes to try and clean it up. The key thing here is that we're filling in the holes and then we're running the watershed. The reason we want to fill in the holes is because if we run the watershed without that, you end up getting over segmentation. It's something you could try. You could try running the watershed on a duplicated image without hole filling, and maybe you'll get a better idea of what can go wrong. Don't worry at this point if we still end up with a few small speckly objects in the background. The reason we took that size filter or the size measurement for the nuclei earlier was so that we can get rid of those at the analyze particles stage. We're just going to test out a couple of different binary operations. Now the first one we could do is to fill the holes. However, here we can see that we don't actually have any holes. So for the sake of generating one, I'm just going to select a small region here, process, math, add, and add 255. So I've created a hole in this one. So obviously you wouldn't normally do that, but just for the purpose of demonstration, we can go to process binary and fill holes, and that has disappeared. The other process we're going to do is to run the watershed. So this has split down a lot of these, however it has missed off this one. And it's kind of the fact of image analysis and automated image analysis like this, that you're never going to get 100% perfect results, so we just kind of have to live with it. We could try and optimize it a bit further. We could also try and use different processes. Like I say, the watershed here is using a distance-based watershed. However, there are more complicated versions using intensity-based watersheds that we could use. However, that's kind of beyond the scope of what we're trying to do here, but it is something you could look up if you were interested. We've now reached the point where we can analyse our particles using the analyse particles function. You'll find this under analyse and analyse particles. What analyse particles does is it identifies contiguous regions of foreground labelled pixels and consider those to be individual objects. The analyse particles dialog looks like this. So here, importantly, we're able to specify an expected size range. Now we know that in microns squared nuclei are around 150 microns squared. So for this, just to put in a very coarse size filter, we might say that we would only accept things in the range 50 to 500, say. You can also specify that these are done in pixel units rather than in microns. However, we know our size measurements are in microns, so you can leave that unticked. And also we've got a circularity threshold here, where 0 would be something, I guess, infinitely straight thin line, and 1 would be a perfect circle. You have additional options here based on what you want to output as images. If you want to output any images at all, you can output overlays, or what's quite useful I find are these count masks. So what counts will give you is each individual object, the pixels in the output image will be assigned to the unique ID number for that object. So let's say this is object number 15, then all the pixels in this object would be number 15. And this provides a nice way for you to go back and identify the same objects in subsequent analysis if you need to. You can also apply that Glasby lookup table when we were looking at lookup tables earlier. What this does is it applies a semi-random color to each intensity level, and it makes it very quick for you to see if adjacent objects have been identified as the same thing. So here, for example, these are very close together, but because they've been identified by, or are being displayed by different colors, we know that they're being detected as separate objects. However, if these all turned up in the same color of green, we'd think that maybe they were being identified as the same object and that we needed to refine our segmentation process. You can also have it automatically make measurements and generate a separate summary table. You can also add them to the region of interest manager as separate regions of interest. So this would be another way that you could then access those regions in the future if you wanted to reanalyze something. And another important feature here is the ability to exclude on edges. If you're measuring things like area, you don't want to include anything which is semi-obscured by the edge of the image because you don't truly know how big it is. So it's useful to exclude on edges. Simply for Worksheet 9, all we're going to do is run the Analyze Particles tool on our binarized image. Don't forget to redirect any intensity measurements to that separate image that we extracted a couple of sessions ago, or sections ago, and let's output them either as outlines or count masks. For Worksheet section 9, we're going to run the Analyze Particles process on our binarized image. Typically what you'd want to do is go to Analyze, Set Measurements, and do, if you were measuring intensity, do redirect intensity measurements to this image. However, for the purpose of demonstration, I'm just going to go Analyze Particles without setting that. I'm going to set our size range here to 50 to 500, knowing that our sort of average nucleus size is around 150. So this is being pretty pretty coarse, but it will remove these small speckly objects. And had we got anything that was absolutely huge, sort of beyond reasonable size, it would get rid of that as well, although there's nothing in here that should. I'm going to exclude on edges, want to display the results, clear any results that have previously been generated, do summarize, and also add to the region of interest manager, might be useful. And I'm going to generate the count masks image. So if I run that, you can see that we've got our results table here. So for each of these, we found 56 objects, and we've got area measurements in microns squared, and the X and Y center coordinates. But importantly, we've got mean, minimum, and maximum intensity values of 0, because we didn't redirect the measurements. So all it's doing is it's identifying the object here and saying that the intensity is 0. So if we remove everything here, discarding all of this, and then go to Analyze, Set Measurements, but now redirect to that intensity image, when I run this again, Analyze Particles, same settings, now it gives me correct intensity information. So for example, for nucleus 34, it's actually taking these intensity measurements from over here. We've got the summary, so this just tells us the number of objects, which image we processed, and then some basic metrics about the size of the image, the size of the objects, and also how much of the image was covered by objects. We've got the objects in the region of interest manager here, so you can see it highlighting on that count masks image, or if we go to this, we can select them here. And we've got this count masks image over here. So as I say, if we go with this object here, so number 34, if we find the corresponding one over here, which we can find, if you look at the context sensitive region of the ImageJ menu, you'll see that the intensity value across that is all 34. And that makes it a nice way for us to go back and identify the same objects. And one last bit is if we go to Lookup tables, and then Glasby inverted, you can see how each of those has been given a random colour. So we can very quickly see that these 3 objects here are identified separately, as were these 2, but this one here was identified as a single piece. With the measurements for our nuclei, we're just going to have a quick look at some of the plotting and fitting tools that are available in ImageJ. From the results window, you have this results menu that we briefly touched on earlier. One of these options is plot. What plot allows us to do is to plot 2 of the columns. In this case, I've gone with area and the mean intensity against one another. You can either do this as connected lines or as a kind of scatter style circles in isolation. If you click the More button here, you'll have additional options for things like label sizes, legends, and exporting the image at a high resolution. And if you click on Data, there's also data fitting, which we'll look at in a couple of slides' time. One of the other things you can do from the results table is to plot the distribution as a histogram. So here we're able to specify the number of bins and what bin range we want to use over. So here I've plotted the area distribution, and in fact this is something we'll be doing as part of the final exercise. If you've got an XY scatter plot like this, you can go to Data and then Add Fit, and that's done from the plot window, and then you'll get multiple curve options. So you could do things like straight lines, multiple order polynomials, Gaussians, exponentials, and a bunch of other plots. When you do fit this, and so I've just gone for a straight line here, you'll see the results displayed in the log window. These are just the fitting parameters. For the final exercise, what we want to do is fit a Gaussian to the distribution of nuclear areas. In order to do this, we're going to have to go through a couple of steps, mainly because the distribution window doesn't have the ability to fit a plot. So first off, we're going to have to generate that distribution. We've gone here, or I've gone with 5 bins from 100 to 200. Then with that distribution window, you're able to get the bin starts and the number of counts per bin. We can then rename that results window to results, which gives us that results menu. In results, we'll then plot the points as 5 separate circles, or however you want to plot it. And then we can use the plot window's fit function to fit the Gaussian and extract out the center of that Gaussian and the standard deviation. So for the final exercise, I've just left us with this results window from exercise 9. This has got the area measurements as well as some of the intensity and position information. It's the area that we're interested in there. So we'll go to results, distribution, and then say plot area, 5 bins, and in the range 100 to 200. So we get this out now. Now there's no ability to fit a Gaussian in this window, so what we have to do is list this out and generate a results table. And as I mentioned previously, This doesn't have that results option like we do here because this window isn't called results. So what we'll have to do is rename this one. So we'll just, results 1 will do, and then rename this one to simply results. So we've got those. The reason for renaming this one first is that otherwise this would overwrite that one and we'd lose all this data. So now with this results table, we're able to use the plot function, and we're going to plot the bin start against the count. Let's do blue circles, and let's make them a little bit bigger. So we can see that. So this just follows the same distributions we had there. They're now just XY scatter style. Click OK. And the final piece we do is go to Data, Add Fit, select Gaussian from down here, and then let's make that red so it stands out. And in the log window, we now have our Gaussian fit. So it's telling us the parameters and how they fit into this Gaussian equation here, and the line displayed over the plot. So before we wrap everything up, I just wanted to go through a couple of extra features and things of ImageJ and Fiji. First of these is the ability to install plugins. You can do this by just dropping jar files into the plugins directory of your Fiji installation. However, it also has this package manager. You can get to it by going to Help and Update, and then if you click on Manage Update Sites here, you'll get shown this window, and this is just a list of all the available plugins which are kind of come semi-bundled with ImageJ, or with Fiji. So you can put a tick next to the ones that you're interested in. So for example, I put a tick next to 3D Script. Then close that window and then just click Apply Changes, and it will download all the relevant bits of sort of plugins and jar files, and when it restarts ImageJ, you will have those functions available in your plugins menu. And finally, one of the cool features of ImageJ is its ability to automate processing using macros. Now macros is a massive topic, and indeed we do actually have a separate course on this, which you can find a link to in the description below. However, just to give you a taster, and also as a bit of a shameless plug, we're going to go through just a kind of example workflow in order to automate what we've been doing today through the worksheet. So a macro is just a list of text commands which automate the different processes that we're running every time we're clicking buttons within ImageJ. To access this list of text commands in real time, you can go to Plugins, Macros, Record, and that will open this window here, and every time we click something, you'll see the command corresponding to that appear here. So for example, when I ran the background subtraction earlier, it did run subtract background, rolling 50 and sliding. So that would be subtract background with a rolling ball radius 50 and the sliding paraboloid method. You can then click create, and this will open up in the script editor where you can edit this, for example, getting rid of sort of specific features such as the explicit names of images, and also just running it. So you could open up an image, click run, and it would rerun that series of processing steps. You could also paste that macro code into the batch processor here. So this would be useful if you've got, say, tens, hundreds, thousands of images, and you can put those into a single folder, and then you select that with input, and then you create a blank folder which you select with output, and then just click process, and for each of those images it sequentially load it in, run the macro code, and then save whichever image is open at the end to the output folder. Okay, so quickly to demonstrate macros, we're going to just run through the same workflow that we've been working through today, but just with the sort of select few steps that we optimized. So we're going to run the macro recorder. Here I just did it by clicking on Dev and Record, or you can go the long way, which is to go Plugins, Macros and Record. So we'll leave that running on the right hand side, just extend it out. And then we'll open in our data here. So first thing is to split those colours out, split channels, remove the second one, then do a max projection. For a bit of variety and simplicity, let's go subtract background, 20 pixel rolling ball, sliding paraboloid, OK. Then do a median filter, 2 pixels, run an auto threshold of Huang, disabling that. Then binary operation, watershed, split them down, so that looks basically the same as we had before. And then we'll run analyze particles. Don't want too much popping up, so let's just display the results and display the count masks. and then with that image we will change the lookup table to inverted, and that's it. So if we click create now, that opens up in the script editor, and we could rerun that, which we'll do now as a demonstration. And close the macro recorder. So I'll reload that image. Okay. And if I click run now, it should automate all of those steps, and we end up with our results table and the segmented nuclei shown with the Glasby. However, what you'll notice in this script is that a few of these have got hard-coded references to the image name. In fact, a lot of them have example-data.lif. So what we need to do is replace anywhere where it says example-data.lif with the name of that image, which we can extract using one of the macro functions. So if I open that, and then obviously this is always still called example-data.lif, so just for the purpose of this, I will rename it to testdata.tif or something. Now if I run this, it's going to fail because it's looking for example-data.lif, and in a second it will just give me a warning, no window with the title example-data.lif found. So instead what I can do is I can use the getTitle function, and I'll just call this name. So using the getTitle function, assigning it to the variable name, and then anywhere where it says example-data.lif, I'm going to replace that with name. So name. Here, use some string concatenation. Now, don't worry too much about all of these different things in terms of like using functions and string concatenations. This is the purpose of the separate macro session that we've got, and again you can get to from a link in the description below. But for now, this is more just a demonstration of what you can do. So here, all it's doing is it's getting the current name of this window. So in this case, now it's testdata.tif. And here, wherever it says name, it's inserting that into the text. So hopefully, if I have done that right, when I click run, boom, 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 and it runs it. So the idea here is that then this would be able to run on any image that was given because it's dynamically getting the name of that window. And that's it. That is the end of the image processing analysis in ImageJ Fiji course. Hope you've enjoyed it and found it useful. If you've got any questions or comments, feel free to pop them in the comments at the bottom of the video, or contact us at the website here, bristol.ac.uk/Wolfson Bioimaging, or you can message us through our GitHub repository. As I mentioned, we've got a couple of other courses. So we've got the macro course. This one we list out as level 2. So this goes through the kind of automation that we've just looked at, as well as some higher functionality. So using functions, and also a separate CellProfiler course. So a completely different piece of software, but again, really useful. We do have a separate course on that. So thanks again, and have fun.